Hello and welcome to BioCode. In this video, we will see how we can utilize the alphabets of BioPython that BioPython provides, and what are the real world use cases of this learning these sort of functionalities from that are provided by BioAlphabet. So basically, in this video, what we are going to accomplish is that we are going to manipulate sequences in a way that we take them as input or read them as files, and then we are going to concatenate them together. For example, let's suppose that you have 10 different files and 10 different sequences that you take as input from the user, and then you would like to add them together into a single sequence. How would you do that? So in this video you will learn that basically we are not going to utilize any files or a huge amount of sequence. We are just utilizing short sequences. So these are just strings rather, and just to make a point of your observation for your observation. So first of all, what really is happening is that we are importing the sequence class, then we are importing the generic DNA, generic alphabet. We also need to import generic protein, and there we go. Then we are also importing the IUPAC class, which we recommend, and then we are declaring a MyDNA object, and we are directly passing the sequence to it, and also saying that it is an unambiguous DNA. There is no ambiguity within our sequence, and then we are actually printing it out as a DNA sequence. Same goes for the protein. We are defining it as IUPAC protein, and then what we are trying to do is we are basically let just disappear. Okay, so basically what we are trying to demonstrate over here is that we would like our DNA sequence to be merged with protein sequence. For example, what we are trying to make a logic is that it should look like this after the result. So my complete sequence should look like this, right? Because there is a concatenation between plus, and Python allows concatenation of strings, right? So let's run this program and let's see if it actually works. So I'm going to go to run without debugging, and you will see an error that says incompatible alphabets. So that means that we cannot really concatenate two different sequences, because we defined a single sequence as a DNA, and then we also defined a second sequence as a protein. So in the natural world, in the real world, we cannot add a DNA with a protein. What is the use of it? And this is not a possibility. So same goes within the computational side. We cannot merge a sequence of a DNA and merge a sequence of protein together and concatenate them. We can obviously convert them into string and then do that, but what is the use of that? So BioPython does not allow you to do that. It will give you a type error that will say incompatible alphabets, and you cannot really merge them together. So how do we go around that? How do we utilize the generic functionality of IUPAC and make them together? So basically what this is the use of generic functionality. Generic DNA, generic alphabet, generic protein, basically it allows us to give a definition to our sequences in a way that they are defined as generic. They are defined as a general sequence. So if we define a protein and if we define a DNA, both of them as general alphabets, we can merge them together, right? So for this to work, we need to define this sequence as a generic alphabet, and then this sequence as a generic alphabet as well. So it will work. Okay, so I actually made mistake over here. It will now work, and you will see that it will be concatenated and then it will work. So now you can see that our sequence has been merged. Basically these results are from the below code, and we will discuss that later. So now you can see that our code has worked. So let's change it into a generic protein, and let's see if it actually works this time. There we go. So once either of them is a generic DNA, generic protein, generic alphabet, you can merge them together. So as long as they are not strictly defined as protein, as a DNA, as an ambiguous DNA, an ambiguous protein, an ambiguous RNA, they cannot be merged. But as long as they are not defined as strictly or restrictly defined as protein, DNA, they can be merged together. So this was a possibility of utilizing them. And let's go down, and now we can, as you can see, we actually defined it underneath as well, but I'm just going to remove this code now, and let's go back. Okay, so what is the use of utilizing learning this? Basically, as I said earlier in the start of the video, that we will be merging together sequences, a chain of sequences. For example, let's suppose that you have three sequences, and basically this is a list of sequences, so please do not get confused in that. This is a list of sequence objects basically, and in that sequence objects we are actually directly defining these sequence of objects. So I think it will be pretty hard to understand, so I'm just going to declare a different my object 1 and 3. So let's define them over here. There we go. So now we have defined three sequence objects over here, and now you can see that all three of them are basically DNA sequences. They are generic DNA sequences, and we have defined their objects. Now we need to put them in a list, and if you don't know how to do that, you need to watch our first videos about the list. And there we go. So our list is ready. You could basically do just